This lesson is about how things enter and leave the cell. We sometimes call that passive transport and active transport. But to understand these things, we need a little bit of background. One of the things we need to remember is what makes a solution. Solution has some kind of solute in it: salt, sugar, oxygen, some kind of solute in it. That solute is being dissolved by a solvent, and together that makes a solution. So in my example, I've got sugar, that's the solute, water is a solvent, and together they're a sugar-water solution. If I add more sugar, it becomes more concentrated. If I add more water, it becomes more diluted. That's important because of what's called the kinetic molecular theory. That is that all molecules and atoms are in a constant state of random motion. So in that solution, that sugar that's dissolved is in a constant state of random movement. It doesn't stay in one location, it's constantly moving around. If we remember, the plasma membrane is a lipid bilayer. That means that there's two layers of lipids and that it's semi-permeable, which means some things can enter the cell, but not everything. And some things can leave the cell, but not everything. So here we have an example of a lipid membrane. We have a high concentration of some solution on the outside and a low concentration on the inside. Because those molecules are randomly moving, they're going to move through the plasma membrane until they reach a point where they're about equal on each side. That doesn't mean the movement stops. The movement continues, but they stay about equal on each side. Dynamic equilibrium. Particles move from an area of high concentration to an area of low concentration. It does not require any energy. Kind of like this kid here on the sliding board. It's going to go from an area that's high to an area that's low, but it doesn't require any energy. Just the force of gravity will allow that slide to work. In a similar way, the more I increase the concentration, it's like the steeper and steeper that slide becomes. So the faster and faster that diffusion process happens. So if I increase the concentration, it just simply means that the diffusion, the random movement of particles, is going to happen faster. Here's an example of some molecules and a semi-permeable membrane. So right now, the water molecules, symbolized by blue dots, are in higher concentration in area of B. So they're going to go from an area of high concentration to low. So the water molecules are going to go from B to A. But the other molecules, we'll call it salt, for example, the salt is in higher concentration on A, and they're going to move from A to B. So just randomly, some of those molecules are going to go from one side of that membrane to the other. And in a similar way, at the same time, the water molecules are going to go from an area of high concentration to an area of low concentration. In this little experiment, we're going to see exactly that. So we put a semi-permeable membrane around a sugar and starch solution. We put that into water with some iodine. We can see how the high concentration of sugar and starch would mean that those molecules are leaving that artificial cell and entering the water. However, the starch is too big of a molecule to fit. The glucose, the sugar, on the other hand, is not. So the sugar will be able to move across that membrane, but not the starch. The water and the iodine can move across that membrane. So inside that cell, the starch is going to turn color because the indicator will mark it. But on the outside of the cell, we'll have sugar, and we'll still have some of that starch indicator. In an odd sort of way, we can also see how this diffusion works in this weird-looking U-shaped tubing. In this case, I have a very high concentration of some solute, like sugar, on the B side, and a low concentration on the A side. As a result, water will move from the A side to the B side. The reason is water was in low concentration on side B, so it's going to move from side A over to side B, which makes it have this really weird looking appearance. We can name these kind of solutions. If the solution has a high concentration of a solute in it, we call it hypertonic. Because it has such a high concentration of the solute, it has a very low concentration of water. In that case, water is going to leave the cell because there's a higher concentration of water in the cell, a low concentration of water in the solution outside of the cell. If there's not much salt or sugar in the solution, it's a hypotonic. So the water is moving into the cell, causing the cell to swell up. So I remember it kind of like hypo swells up like a hippo, like an O, a big O. Whereas hyper means it's going to shrivel up. The cell shrivels up. Isotonic just means it's the same. So there's the same concentration of water and solutes both inside the cell and out. So things like saline solution, contact solution, those are isotonic solutions. So take a look at these three, pause the video and see if you think the solute will diffuse in or out, and if you think the water will diffuse in or out. Water cannot just move through the membrane. It needs help. And the reason is because oil and water doesn't mix. Those two lipids on the inside of that bilayer won't allow the water to pass through it. So special proteins that channel through the membrane allow water to enter or leave. These are called aquaporin. All right, so the protein ends with IN, so aquaporin, and the water is coming into and out of the cell through that channel. Active transport just means that the cell has to expend energy. It's going to go against the concentration gradient. Instead of from high to low, in this case, it's going to go from low to high, but energy is required. So here's an example where we're going to expend some ATP. We have a membrane protein that is going to use that ATP in order to pump some molecules into the cell, even though it's against the concentration gradient. Alternatively, they might pump molecules out of the cell, even though it's against the concentration gradient, but it requires energy. This process is called endocytosis. It's where the cell swells up and engulfs some particles. So like white blood cells do this with bacteria. And because it's going into the cells, endocytosis. The reverse process of the cell manufacturing something and then releasing it out of the cell is called exocytosis, like exiting from the cell. So either way, this also is going to be a form of active transport because some kind of energy is required. So we have a passive transport, no energy required. We have active transport where there is some energy required. There's also facilitated diffusion, like for example, the protein helping the water to go in and out. Now, it's facilitated because it doesn't just happen randomly. The protein channel is allowing that water to enter, but there's no energy required. I hope this video helps describe different types of transport across cell membranes.